Good afternoon, everyone, and welcome to our third webinar. My name is Myriam Croze, and I will do this webinar with Véronique Giudicelli. Today, we will present the axis 2 of IMGT, focusing on the analysis and exploration of the expressed immunoglobulin and T-cell receptor repertoire with IMGT tools. In axis 2, we analyze rearranged DNA and RNA sequencing data using tools based on the reference directories from axis 1. This process allows us to identify and annotate V-domain nucleotide sequences and analyze clonotypes. Here are listed the IMGT tools available in axis 2, which we'll explore in detail today. I will introduce you to IMGT-QUEST and its integrated tool IMGT JunctionAnalysis. Véronique Giudicelli will cover its high-throughput version, IMGT HighV-QUEST, as well as IMGT StatClonotype. This graphical representation, or prototype, illustrates rearranged immunoglobulin and T-cell receptor V-domain sequences with IMGT labels at the nucleotide level. There are two types of genes rearrangement. The first involves the V, D, and J genes, resulting in the VDJ region. This rearrangement is observed in the immunoglobulin heavy chain and the T-cell receptor beta and delta chains. The second rearrangement occurs between the V and J genes, forming the VJ region, and is observed in the immunoglobulin light, lambda and kappa, and T-cell receptor alpha and gamma chains. IMGT's unique numbering for immunoglobulin and T-cell receptor V-domain delimits the four framework regions, FR1, FR2, FR3, and FR4 IMGT, and the three hypervariable or complementarity determining regions involved in the antigen binding, CDR1, CDR2, and CDR3 IMGT. The CDR1 and CDR2 IMGT are encoded by the V region, whereas the CDR3 IMGT results from the VDJ or VJ rearrangement. The junction encompasses the two constant amino acids, the second cysteine at position 104 and the J-tryptophan or J-phenylalanine at position 118, with the CDR3 between including N region from the N diversity. IMGT-QUEST is an online tool which allows you to analyze up to 50 nucleotide rearranged sequences of immunoglobulin and T-cell receptor per run from genomic DNA or cDNA data. This tool compares user sequences with IMGT reference directory sets for the identification of V, D, and J genes and allele, the evaluation of mutation and amino acids changes, the analysis of the junction with the incorporated tool IMGT JunctionAnalysis, and finally the full annotation of the V-domain. To demonstrate how IMGT-QUEST works, I will use an example of three rearranged RNA sequences from human immunoglobulin heavy chain transcript. You can see these three sequences here. These sequences will be provided in addition to this video if you would like to run them later. First, I will open the web page of IMGT by clicking on this link. Here you might recognize the web page of IMGT that has been presented during the first webinar. It features tools from the three IMGT axes that I presented here, and we can access IMGT-QUEST tool by clicking on its name. Here you can see the welcome page of IMGT-QUEST. On the top are listed reference articles on these tools. Under are provided two links. The first gives access to the list of the IMGT-QUEST reference directory sets, to which users' own sequences can be compared. And the second provides examples of human rearranged sequences to test the tool. The page then includes several sections to configure the analysis. In the first section, called Your Selection, you must select the species or taxon that you are studying. For example, Homo sapiens, a human. Then you have to select the receptor type or locus in this list. In the sequence submission, you can enter your nucleotide sequences in the text area in FASTA or FASTQ format. Alternatively, you can upload the sequences as a text file by selecting this option. Below, the Display Results section offers 3 displays for viewing the results: a detail view and synthesis view, which can be displayed online in either HTML or plain text format, an Excel file which is designed for downloading the results. Both the detail view and synthesis view include sequence alignment, where users can specify the number of nucleotides per line. The default value is 60 and you can go up to 10,000. In the detail view, you can adjust the number of IMGT reference sequences aligned with the user sequence from 1 to 20, and the default is 5. This display will give results for each sequence individually, including a summary of the analysis and 14 detailed results sections, which are listed here, and can be selectively checked or unchecked by the user. If you select the synthesis view, it will group sequences expressing the same V gene and allele. The results include a summary table, which can be sorted either by V gene and allele name or by the sequence input order. It will also give you 8 detailed results sections listed here. Finally, selecting Excel files allows you to download the results either as a spreadsheet or as a zip archive, and the results can include up to 12 sheets listed here. Users can also display the content of a specific sheet directly in the browser, or download the results in R formatted form, which is another format specific to immunoglobulin and T-cell receptor. The next section is called Advanced Parameters. The default values are used by IMGT VQuest for classical analysis, however, they may be modified for specific studies and/or unusual sequences. The user may select the IMGT reference directory set to include. For example, you can include pseudogene or not. You can include all alleles or only the allele 01. You can search for insertion and deletions in the V region. You can set parameter values for the integrated tool IMGT JunctionAnalysis, which analyzes only the junction. These parameters include the number of accepted digits, with the default value indicated in the bracket. You can also set the number of accepted mutations in these 3 regions. Then you can also set parameters for the detail view, as for example the number of nucleotides to exclude in 5' of the V region for the evaluation of the number of mutations, or the number of nucleotides to add or exclude in 3' of the V region for the evaluation of the alignment score. Finally, the advanced functionalities allow you to analyze specific sequences tailored for single chain fragment variable SCFV, which is designed for sets containing engineered single chain with 2 V domains connected by a linker, or clinical application to search for subsets from patients with chronic lymphocytic leukemia. I will now submit the 3 previously mentioned sequences and run the analysis of IMGT VQuest. For this analysis, I will search for insertion and deletion in V region. I will display the results in detail view. For the alignment, I will have 150 nucleotides per line. The species we are analyzing is human, and it's the locus immunoglobulin heavy chain, and I will copy the 3 sequences in the text box. And I will now start the analysis by clicking on Start. Here you can see the web page with the results of IMGT V-QUEST. On the top, you have a summary of the selected parameters. Under is indicated the numbers of analyzed sequences, in our case 3, and the list of sequence identifiers with link, allowing to browse directly the corresponding individual results. From there, you will have the results of the first sequence analyzed. The sequence length is 633 base pair. The sequence was analyzed under category 2 for indel search and correction, and the sequence was compared with the Homo sapiens immunoglobulin set from the IMGT reference directory. The FASTA sequence is also provided with the V domain highlighted in green. Under, you will see the result summary table that outlines the main characteristics of the analyzed sequence. The sequence functionality is given in the first row. Our sequence is productive, with no stop codon, and the junction is in frame. If either condition were not met, the sequence would be unproductive. The names of the closest V and J genes and allele are provided with the alignment score, the percentage of identity, and in bracket, the ratio of the number of identical nucleotides to the number of aligned nucleotides. For example, this sequence expresses the V gene IGH V439 with an identity of 96.91% to the reference sequence. The name of the closest D gene and allele determined by IMGT JunctionAnalysis is also indicated with the D region reading frame. In this line are described the length of the 4 FR IMGT and the 3 CDR IMGT, as well as the amino acids junction. For example, this sequence has a length for the 3 CDR IMGT of 10, 7, and 17 amino acids. Finally, the last row indicates the junction length, which is 57 nucleotide, and the junction decryption that I will explain in more detail later. Below, the selected result section of the detail view will be displayed. In the first section, the alignment and identification of V genes and alleles are presented. A list of the 5 closest germline V genes and alleles is provided, along with their alignment score and percentage identities. Below the list, the alignment of the analyzed sequence is displayed at the top, compared to the 5 genes and alleles. Hyphens indicate identical nucleotides between the analyzed sequence and the germline genes and alleles, whereas letters represent mismatched nucleotides. Dots denote gaps in the alignment that are the missing nucleotides based on the IMGT unique numbering. Finally, IMGT labels are also given on the top. You will also get similar results for the J gene and allele identification. In the next section, you have provided the results of IMGT JunctionAnalysis with here the default parameters. You will get the analysis of the junction that describes the junction at the nucleotide level with IMGT defined region, including the 3' V region, the N1 and N2 regions that result from the N diversity, the D region, and the 5' J region. In this case, dots are used to indicate the number of nucleotides trimmed at the germline ends of the V, D, and J genes. Mutations in these regions are identified and noted as VMUT, DMUT, and JMUT, with the corresponding nucleotides underlined in the sequence. For example, you have 2 mutations in the D region, and the original reference can be viewed by clicking on it. For instance, if I click on the T, you can see that the original nucleotide is a A. You will also get the junction description that will describe the length in nucleotide of the IMGT labels of the junction. For example, in this case, the 3' V region consists of 11 nucleotides with no nucleotide trimmed. The N1 region has 4 nucleotides. Then the D region spans 20 nucleotides with 1 nucleotide trimmed in 5' and 10 nucleotides trimmed in 3'. The N2 region contains 4 nucleotides, and the 5' J region has 18 nucleotides, including 3 trimmed. The analysis also includes a translation of the junction in amino acids. This translation shows the amino acid junction sequence. The junction frame is given. In this case, the plus means that is in frame. You will get the CDR3 IMGT length, which is 17 amino acids in this example. And any amino acid changes are also underlined, and the original reference can also be viewed by clicking on it. For example, if I click on the phenylalanine, you can see that the original amino acid is a tyrosine. In the section 5 is provided the junction sequence in nucleotide and amino acid, as well as a sequence in FASTA format with the formatted header required as input by IMGT JunctionAnalysis tool. In this section, you have the V-region alignment according to the IMGT unique numbering for the nucleotide sequence analyzed, along with the 5 closest germline V genes and alleles, with the FR IMGT and CDR IMGT delimitation. In the section 7 is given the nucleotide sequence and its amino acid translation aligned with the closest V gene and allele. In the section 8, the V-region amino acid translation of the input sequence is aligned with the closest V gene translation, with the amino acid changes shown in bold on the third line of the alignment. The next 3 sections focus on analyzing the nucleotide mutations and amino acid changes observed in the V region compared to the closest germline V gene and allele. In this table is presented the nucleotide mutation and for non-silent mutation, the corresponding amino acid changes. These mutations and changes are described for each FR and CDR IMGT region, with the nucleotide and codon position provided according to the IMGT unique numbering system. In addition, the amino acid class change type, shown in parentheses, indicates whether the hydropathy, volume, and physicochemical properties of the replaced amino acid are constant, indicated by a plus, or replaced, denoted by a minus. For example, in the CDR1 IMGT region, you have 2 mutations that result in amino acid changes. You can see that at the position 103, you have a mutation from A to G, with an amino acid changes from a serine to a glycine. And you can see that also in this alignment indicated here at the position 35. In the next section, you have the V region mutation and amino acid change statistics. It includes 2 tables. The first table, called the nucleotide mutation table, quantifies the nucleotide positions, including those with or without gaps, the number of identical nucleotides, the total number of mutations, and the number of silent and non-silent mutations, both for the entire V region and for each FR and CDR IMGT region. The table further breaks down the analysis by the 4 types of transition and the 8 types of transversion. Secondly, you have the amino acid changes table that quantifies the amino acid positions, both with and without gaps, and lists the unchanged amino acids as well as those that have changed. The table provides a breakdown of the amino acid changes for the V region and for each FR and CDR IMGT region. Additionally, it evaluates the degree of similarity for each amino acid change, categorizing them into 4 groups: very similar, similar, dissimilar, and very dissimilar, based on the number of physicochemical properties conserved or not. The V region mutation hotspot section highlights the location of mutation hotspot in the germline sequence indicated in this table. It also presents a table similar to the one shown before, but with the added feature of identifying the hotspot mutations. These mutations are indicated within square brackets, as for example in these 2 mutations in the CDR1 IMGT. The section 12 provides nucleotide and amino acid FASTA sequences with gaps of the V region as well as the VG or VDJ region. In this section, you will get the full annotation of the VJ or VDJ region by IMGT Automat, and it is provided with IMGT labels. Finally, in the last section, you will get the IMGT collier de perles that has been described in the first webinar. You can visualize the collier de perles by clicking on this link. Here, you can visualize the collier de perles with each CDR indicated in different color, and this circle here indicates the gaps in the CDR. The results of the second analyzed sequence are shown here. In the summary table, you can see that you have the presence of indels in the sequence. There is an insertion of a single nucleotide C in the FR3 IMGT at codon 83 of the V region, corresponding to position 374 of the submitted sequence. Additionally, one deletion is also observed in the CDR2 IMGT region. Both the insertion and deletion lead to frame shift. However, IMGT-VQuest considers indels as sequencing error and automatically reanalyzes the sequence, removing the insertion and marking the deleted nucleotide with a dot. You can also see a warning that appears as note in red under the table, indicating that IMGT junction analysis gives no results. Other warnings can be displayed to alert the user. The list of the warnings can be found in the documentation. I will now pass the detailed results and show you the results for the third analyzed sequence. Here are the results of the third analyzed sequence. In this sequence, I wanted to show you a case where we have an incomplete V region. In this case, this will be indicated by the text V region partial in 5' missing nucleotide number equal 20. You can also see the missing region in the V gene and allele alignment below. This will end the presentation of IMGT-VQuest. If you want to go further, you can have a look to this link, as for example the IMGT-VQuest documentation or the list of warnings. Véronique Giudicelli will now present IMGT HighV-QUEST, the high throughput version of IMGT-VQuest.
Thank you, Myriam, for your introduction. Let's now change scale and focus on the analysis of thousands of round sequences of immunoglobulins and T-cell receptors resulting from the high-throughput sequencing in order to capture, at a given point, at least a partial view of the expressed repertoire. I will present the two modules of IMGT HighV-QUEST. The first one, corresponding to IMGT V-QUEST for the sequence analysis, and the second one, statistics, performs the clustering of sequences into clonotypes according to the IMGT definition in order to set immunoprofiles. We will have an overview of the IMGT StatClonotype tool, which compares two immunoprofiles resulting from the IMGT HighV-QUEST statistical modules. IMGT HighV-QUEST is a high-throughput version of IMGT V-QUEST. It is on the web since 2010. The sequence sets are submitted through a web interface. The tool is freely available to academics and licensed to private companies. The registration of users is required. This allows the tool to automatically notify them by email when the results are available. IMGT HighV-QUEST can process sets of up to 1 million sequences formatted in FASTA or FASTQ. It comprises two modules. The sequence analysis module provides the results in CSV files. These results correspond exactly to those of IMGT V-QUEST when the Excel file output is selected. The statistical module classifies the sequences into clonotypes in order to generate immunoprofiles. Moreover, it can compare the results of several sets for the identification of common clonotypes. Let's see now how to use the tool from the IMGT homepage by clicking on its name. Here is the login page of IMGT HighV-QUEST. New users can register through this link, and once your account is activated, you can log in to access the main page of the tool. Note that the complete documentation of IMGT HighV-QUEST is available by clicking on its name on the top of the page. Use this page to submit your sequence sets. For each set, you will have to provide a title, to select the species and the receptor type or locus, and then to upload a text file containing your sequences formatted in FASTA or FASTQ. By default, you will be informed when the analysis is completed. However, you may also ask for a notification when the analysis is queued on the IMGT servers. Regarding the results, by default, they are provided in 11 or 12 CSV files. You may prefer to obtain them in a single file, then choose the R format, or you can obtain both formats in the results. For sequence sets of less than 200,000 sequences, you may also include in the results the equivalent of the IMGT V-QUEST detailed view results in text format. However, in that case, the analysis will be much longer. Then, you can customize the advanced set parameters as for IMGT V-QUEST. Please note that IMGT HighV-QUEST looks for the insertion and deletion by default. And then you can also select the analysis of SCFV sequences. I remember you that these SCFV sequences are composed of two V domains. Once the parameter selected, click on Start to launch the analysis. You will see then appear the analysis history page. It allows the user to list its own submitted analysis and to check their status, queued, running, or completed, and to remind the submission date, the number of submitted sequences, the species and receptor types used for the analysis, and the actions that can be performed. When analysis is completed, the user can download the result as a single archive file in TXZ format. The availability of the results is guaranteed for 2 weeks after the analysis is completed. After that, the files can be removed by the system. In that case, file removed is indicated in red instead of the download logo. A user may delete an analysis at any time, except if it is used by the second module, statistics of IMGT HighV-QUEST. In such case, use by statistic is indicated in place of the delete button. To illustrate the content of the 11 or 12 CSV files that compose the classical results of IMGT HighV-QUEST, I choose to refer to the IMGT VQuest documentation related to the Excel outputs, since both contain the same data. The available documentation can be reached through this link, and this table indicates for each file its number, its title, the number of columns it contains, and the main data it includes. You can access the exhaustive list of the column of a given file just by clicking on its title. In a more global way, the first file corresponds to the IMGT VQuest detailed view result summary table with the VJG chain and allele identification, the sequence functionality, the amino acid junction, the F4 and CDR lengths, the description of the insertion and deletions, and the display of the warnings. The files 2 to 5 correspond to the annotation by IMGT Automat with the delimitation and the positions of the F4, of the CDR, and of other IMGT labels in nucleotides or amino acids with or without IMGT gaps. The file 6 provides the detailed annotation of the junction performed by IMGT JunctionAnalysis. Then the files 7 to 10 are dedicated to the description of the mutations in the region, with a description of all nucleotide mutations and amino acid changes for each sequence, with statistics for nucleotide mutation and amino acid change, and the list of the hotspots in the closest germline V gene and allele. The file 11 corresponds to the parameters used for the analysis. And finally, the file 12 is dedicated to the analysis of SCFV with the association of the main results of the 2V domains. If you selected the R format for your results, the archive includes the VQuest R TSV file. This file mainly contains IMGT HighV-QUEST results formatted according to the rearrangement schema of the R community. The full content of the VQuest R file is also available in the IMGT VQuest documentation through this link. In this table, you will find the R column names with the correspondence with the classical IMGT HighV-QUEST output, the number and the name of the CSV file, and the name of the corresponding column for the data, with or without additional comments. The VQuest R file also includes fields and IMGT data that do not have an equivalent in the R rearrangement schema. These fields are highlighted in yellow at the bottom of the table, and just to give an example, this is the case of the junction decryption. Following the analysis of sequence sets with the first module of IMGT HighV-QUEST, we need to introduce additional standards before using the second module, statistics. First of all, IMGT has defined the IMGT clonotype AA for amino acid, for the clustering of the sequence. An IMGT clonotype AA corresponds to a unique rearrangement characterized by the names of its V and J gene and alleles, by the sequence of the CDR free in amino acid, and by an in-frame junction. Besides the definition of the IMGT clonotype based on the amino acid sequences of CDR3, IMGT has also defined the IMGT clonotype NT for nucleotide. This corresponds to a unique rearrangement characterized by the names of the V and J genes and alleles, a unique CDR3 in nucleotide sequence, and an in-frame junction. At this level, it is important to note that different clonotypes defined at the nucleotide level can correspond to the same clonotype defined at the amino acid level. Therefore, based on these definitions, we can now also define immunoprofiles for the V, T, and J genes and alleles identified by the analysis of the sequence sets. First, regarding the diversity of the IMGT clonotypes, immunoprofiles will be built by the evaluation of the number of different clonotypes for each V, T, and J gene and allele. And secondly, regarding the expression of the IMGT clonotypes, immunoprofiles will be built by the evaluation of the numbers of sequences assigned to clonotypes per V, T, and J gene and allele. Note that the assessment of the diversity and the expression could be helpful to find clues for a potential clonal extension. The evaluation of the IMGT clonotypes AA and T and of immunoprofiles for the diversity and the expression per V, T, and J gene and allele is the aim of the second module of IMGT HighV-QUEST called Statistics. In order to use the statistical module of IMGT HighV-QUEST, you can click on the link Launch Statistics, which is available on the top of the IMGT HighV-QUEST web pages. For launching a statistical analysis with IMGT HighV-QUEST, you have to provide a title for this analysis, to indicate if you want to be informed by email when the analysis is completed. This is our choice. And you can also provide some comments, for example, to recall the aim of this current statistical analysis with respect of the IMGT rules. Importantly, you will have also to say yes or no if you want that the comparison of the different batches composing the analysis will be effective, and if you want to obtain the list of common clonotypes. Then you have to define the list of batches to be processed. A given batch corresponding to the results of an analysis of a sequence set. Click on the Show button to display the list of sequence analyses which have been completed, and select one or more, and give a short name. Add it to the list to see it in the table. Up to 15 batches can be defined. In our example, we have a list of 3 batches that will be processed to set the list of clonotypes and immunoprofiles, and that will be compared together in order to identify the common clonotypes. Click on Start to launch the statistical analysis. Then the statistics history page is displayed. It allows to follow the status of the submitted statistical analysis and to download the results once completed. The statistical output is provided as a zip file, and we will have a look on its organization and see some examples of its content. The detailed description of the statistical results is available in the IMGT HighV-QUEST documentation through this link. Once the archive extracted, the main folder contains several directories and importantly, the data directory contains text files including the list of IMGT clonotype AA per batch and per chain type with their characteristics in CSV format. And note that these files are required as input by the external IMGT StatClonotype tool that we will see later. You may open the Open to Start HTML file to browse the results through a web interface, and we will focus on the detailed results per batch and on the IMGT clonotype AA comparison. The directory containing the detailed results per batch and per chain type provide several displays of the list of the IMGT clonotype AA and G and the display of the amino profiles as histograms. Let's take the example of the IMGT clonotypes evaluated from the analysis of a human TRB sequence set. The IMGT clonotype per number displays the TRB clonotypes in a table according to their number of associated sequences in the decreasing order, with a link to the representative sequence selected by the tool and another link to get the associated sequences in FASTA format. Another example of detailed results per batch and per chain type obtained in that case from a human IGH sequence set are these immunoprofiles for the diversity and the expression of the V, D, and J genes, which are displayed as histograms with normalized values in order to facilitate potential comparison with other immunoprofiles. In the directory dedicated to the IMGT clonotype comparison, you will find per chain type several files for the characterization of the clonotypes common to 2 or more batches. Full results file lists the common clonotype with the V, D, and J gene and allele, and with the amino acid sequence of the CDR3. The synthesis table provides the numbers of the common clonotypes between 2 or more batches, and then you will find additional files that give the similar results per V, D, and J genes. In this slide is displayed an example of synthesis table. The clonotypes of 3 batches were compared, and first in the table are listed the number of clonotypes that are unique to each batch, with the number of the associated sequences. Then we have the number common to 2 batches with the associated sequences, and finally to the 3 batches. This closes the presentation of the results of the IMGT HighV-QUEST statistical module. The last IMGT tool we would like to introduce is IMGT StatClonotype. This tool is complementary to IMGT HighV-QUEST statistical module as it compares and evaluates the differences between 2 immunoprofiles represented by 2 sets of clonotypes on a statistical point of view, and this for the diversity and the expression of the IMGT clonotype AA. The tool is available from the IMGT ONS page as a standalone package, and the procedure for the installation of the R program and the StatClonotype package is described in the documentation online, and this will allow the user to run the tool in their web browser. Here is the welcome page of IMGT StatClonotype. First of all, upload in the left panel the 2 sets of clonotypes to be compared, and please remind that these sets are selected in the data directory of the output of IMGT HighV-QUEST statistical module, and then adjust the range of the CDR3 length in order to eliminate outliers if needed. Then, the list of selected clonotypes in the two sets are displayed in the two first tabs of the right panel, 1 and 2. IMGT StatClonotype uses a generic statistical procedure for the identification of the significant differences in proportion for the IMGT clonotypes of the two sets per V, D, and J genes and allele regarding the diversity or the expression. This evaluation can be browsed in the tables of the statistical test result tab here. 7 procedures have been implemented in the tool in order to adjust the p-value. This will allow the user to choose the stringency which is the most relevant for the aim of his study. The statistical analysis is also displayed in the multiple testing procedure plots, which comprise also additional graphical displays. The Synthesis Graph tab displays a graphical comparison through the synthesis graph that combines normalized proportions in set 1 and 2 and differences between proportions with significance and confidence interval for genes at the top of the page and for alleles in the graphic at the bottom of the page. The tool also provides, in the CDR length tab, a graphical comparison of set 1 and 2 per CDR length, here CDR free length, of the distribution of the number of clonotypes for the diversity or of the number of sequences assigned to clonotypes for the expression evaluation. The tool displays, in the CDR IMGT AA Properties tab, the distribution of the amino acids in CDR IMGT, here CDR3, for a given length, and here for the set 1, and below for the set 2. Finally, the VDJ Gene Association tab displays interactive heatmaps that represent the association of the V and G gene and alleles in the set 1 and set 2, and it is also available for the VD association as well as the VDJ association. Well, this is the end of the overview of this comparison of sets performed by IMGT StatClonotype. For more detailed information, please consult the documentation of IMGT StatClonotype. To conclude, we had an overview of the IMGT tools dedicated to the analysis of immunoglobulins and T-cell receptor rare sequences. These tools implement the IMGT standards for the sequence interpretation and comparison. They were developed for extensive studies of Ig and TR repertoires, with the evaluation of the V, D, and J gene allele usage, with a detailed analysis of the CDR3 in terms of length distribution and amino acid composition. They provide an exhaustive analysis of the mutation in the region, and they also evaluate the diversity and the expression of the IMGT clonotypes. These tools are used to set immunoprofiles of the immunoglobulin and T-cell receptors for the comparison of B-cell and T-cell populations in normal and pathological situations. They are also used for the study of combinatorial libraries for the discovery of new specificities. We thank you for your attention, and we will be pleased to answer your questions.